Welcome to an in-depth exploration of class Cestode, commonly known as tapeworms, presented by Dr. Indu Yadav. Let's begin with Taenia solium, a species of tapeworm. Here, we see its classification, habitat, structures, and body wall, including the scolex with its hooks and suckers. Class Cestode is divided into two subclasses, Cestode and Cotyloda. This slide compares their key features, including the scolex, genital pore, egg, and intermediate host. Cestode consists of two subclasses, Cestode and Cotyloda. These worms are ribbon-like and segmented, giving them the common name tapeworms. Tapeworms are hermaphroditic endoparasites, ranging from a few millimeters to several meters in length. They lack body cavities and major organ systems, relying on saprozoic nutrition. The scolex, or head, of a tapeworm is globular and equipped with suckers or bothria for attachment. These structures vary between Cestode and Cotyloda. The scolex may have a protrusible structure called a rostellum, which can be armed with hooks. The neck follows the scolex and is unsegmented. The body, or strobila, follows the neck and consists of segments called proglottids. These segments contain the tapeworm's reproductive organs. The central part of the nervous system is located in the scolex, with nerve cords running posteriorly. The reproductive system includes testes, ovaries, and genital pores. In Cestode, the genital pore opens laterally, while in Cotyloda, it opens ventrally. Gravid segments in Cestode detach and release eggs through a process called apolysis. Cotyloda tapeworms have a separate uterine pore for laying eggs singly. Some species exhibit euapolysis or anapolysis, where gravid segments move outside the host. The life cycle of tapeworms involves eggs that can be embryonated or unembryonated. These eggs develop into larvae with invertebrate or invertebrate hosts. In Cestode, eggs hatch within the intermediate host, developing into larvae. In Cotyloda, eggs hatch in water, releasing larvae known as chorocydium. Next, we will explore the stages of bladder worms, also known as metacestodes. Cysticercoid is a type of larval tapeworm found in invertebrate hosts, except for Taeniidae and Mesocestoidae. It has a non-invaginated scolex. Cysticercus is a larval stage with an invaginated scolex filled with fluid. Examples include Cysticercus cellulosae and Cysticercus bovis. Coenurus cerebralis is a large bladder containing multiple invaginated scolices. It is the larval stage of T. multiceps. Hydatiid cysts are large bladders containing brood capsules, where scolices develop. These cysts can float freely in the fluid and are the larval stage of Echinococcus granulosus. Strobilocercus has a single scolex that is not invaginated and is attached to the bladder by a segmented neck. It is the larval stage of T. taeniiformis. Tetrathyridium is the larval stage of Mesocestodes lineatus, characterized by an elongated body and deeply invaginated scolex. The chorocydium is a ciliated embryophore, and the procercoid stage occurs in the first intermediate host. The procercoid is a solid body with hooks on its posterior region. The plerocercoid stage occurs in the second intermediate host, is elongated, solid, and has a scolex like in adults. Cyclophilidae have a holdfast organ known as suckers, which can be armed or unarmed and are 4 in number. Segmentation is present, and the genital pore is on the lateral aspect. Eggs are embryonated when laid and hatch out within the intermediate host. An example is the Taenia egg, which requires only one intermediate host. Apolysis is present. Cotyloda have a holdfast organ known as bothria, which are 2 longitudinal weak muscular grooves. Segmentation is absent, and the genital pore is on the ventral aspect. Eggs are unembryonated, operculated, and hatch out in the water. An example is the Diphyllobothrium laudum egg, which requires 2 intermediate hosts. Apolysis is absent. Tapeworms of ruminants include Monoesia expansa, Monoesia bainadeni, Avitellina lahoria, Stylesia globipunctata, Thysanosoma actinoides, Avitellina centripunctata, and Ithanaesia giardi. Monoesia expansa adults can grow up to 6 meters in length, with a wide scolex and prominent suckers. Segments are wider than long and contain double sets of reproductive organs, known as the double-pored ruminant tapeworm. The vitelline glands form a ring on either side, and the genital pore is marginal. Eggs are triangular and contain an oncosphere or hexacanth embryo with a piriform apparatus. Monoesia bainadeni has a morphology similar to Monoesia expansa, but the interproglottal glands are concentrated at the center of the segment. It commonly occurs in cattle and has square-shaped eggs. Orobotid mites act as the first intermediate host for the life cycle of these tapeworms. Eggs are passed in the feces of the host and ingested by grass mites, where they develop into cysticercoid in 4 months. The final host acquires infection by ingesting infected intermediate hosts, with a prepatent period of 37 to 40 days. Monoesiosis commonly occurs during November and December and is prevalent in young lambs and calves. In adult animals, the infection is mild, but in young animals, it can cause malnutrition, leading to reduced wool and meat production. It also predisposes lambs to bacterial infections like enterotoxemia caused by Clostridium perfringens type D. Diagnosis involves fecal examination for the presence of eggs and gravid segments that look like cooked rice grains. Treatment options include praziquantel, niclosamide, albendazole, and fenbendazole. Control measures include eliminating grass mites on pasture by plowing and cultivation and deworming animals in early summer. Avitellina lahoria commonly occurs in sheep and goats, residing in the small intestine. Adults are 3 meters in length, with short and cylindrical segments. The genital pore opens irregularly alternately, and the uterus resembles a snail shell. The life cycle involves pisocid mites as intermediate hosts. Stylesia globipunctata commonly occurs in sheep, cattle, and goats, residing in the small intestine. Adults are 60 centimeters in length, with segments containing a single set of reproductive organs. The genital pore opens irregularly alternately, and the uterus is dump bell-shaped. Eggs are passed into 2 par uterine organs and have no piriform apparatus. The life cycle involves orobotid mites. Worms are mainly attached at the junction of the duodenum and jejunum. The immature worm penetrates the mucous membrane, forming nodules in the intestine. Heavy infection can cause death. Treatment involves praziquantel at 150 mg/kg body weight. Tapeworms of equines include Anaplocephala perfoliata, magna, and Paranaplocephalomammalana. They belong to the family Anaplocephalidae and are found in the small and large intestines. Anaplocephala perfoliata adults are 5 to 8 centimeters in length, with a scolex that has lappets behind each sucker. Segments are wider than long and contain a single set of reproductive organs. The genital pore is marginal. Orobotid mites act as the first intermediate host for Anaplocephala perfoliata. Eggs are passed in the feces of the host and ingested by orobotid mites, where they hatch and develop into cysticercoid in about 4 months. The final host acquires infection by ingesting infected mites, with a prepatent period of 4 to 6 weeks. Anoplocephala perfoliata is a common tapeworm of horses. Light infections cause no clinical signs, but heavy infections can cause ill health, unthriftiness, and even death. Worms can cause partial occlusion of the ileocecal orifice and perforation of the intestine. Treatment includes mebendazole, niclosamide, and bithionol. Anoplocephala magna and Paranoplocephala mammillana are other species of equine tapeworms. Anoplocephala magna has a scolex with lappets and is 80 cm long, while Paranoplocephala mammillana has a narrow scolex and is 4 cm long. Lappets are absent in Paranoplocephala mammillana. Common tapeworms affecting poultry include Divania proglottina, Railiotina tetragona, Arachinobothriida, Cestode, Cotugnia digonopora, Coanotinia infundibulum, and Hymenalepis carioca. Divania proglottina, the dwarf tapeworm of poultry, affects chickens and pigeons and resides in the duodenum. The worms are microscopic, with a retractable rostellum armed with hammer-shaped hooks. Each segment has a single set of genital organs, and the genital pore opens irregularly alternately. Eggs contain a single egg capsule. Amoeba Tinea sphenoides affects chickens and resides in the small intestine. The worms are small, with an elongate triangular shape. The rostellum bears 12 to 14 hooks, and there are about 20 proglottids. The uterus is sac-like and slightly lobed. Railiotina scolex is a genus of tapeworms that infects birds and mammals. The scolex, or head, of these tapeworms is equipped with hooks and suckers for attachment to the host's intestine. Railiotina tetragona is a common poultry tapeworm that infects chickens, pigeons, and guinea fowl. It resides in the posterior half of the small intestine and uses ants as intermediate hosts. Adults can grow up to 25 cm and have a rostellum with hooks for attachment. Each segment contains reproductive organs, and egg capsules contain 6 to 12 eggs. Railiotina echinobothrida primarily infects chickens and turkeys, residing in the small intestine. It uses ants as intermediate hosts and is known to cause nodular toniasis. The scolex is larger compared to our tetragona, with a rostellum armed with 2 rows of hooks. Each segment has a single set of genital organs, and egg capsules contain 6 to 12 eggs. Railiotina cestode infects chickens and resides in the small intestine, using dung beetles as intermediate hosts. It is usually 4 cm in length and has a large rostellum with 400 to 500 small hooks. Each segment contains a single set of genital organs, and each egg capsule has a single egg. Cotugnia digonopora, also known as the double-pored poultry tapeworm, infects chickens and resides in the small intestine, using ants as intermediate hosts. The rostellum is armed with 2 rows of hooks, and each segment contains 2 sets of genital organs. Egg capsules contain a single egg. Coanotinia infundibulum infects chickens and turkeys, residing in the upper half of the small intestine. It uses house flies and dung beetles as intermediate hosts. The rostellum is armed with 15 to 20 rose thorn hooks, and each segment contains a single set of genital organs. The sac-like uterus contains eggs with filaments. Hymenalepis carioca infects chickens and resides in the small intestine, using dung beetles and flower beetles as intermediate hosts. The rostellum is armed with spanner-shaped hooks, and each segment contains a single set of reproductive organs. Eggs are covered with 3 layers and are rugby ball shaped. Hymenalepis lanceolata infects ducks and resides in the small intestine, using aquatic crustaceans as intermediate hosts. Its morphology is similar to H. carioca. The life cycle of poultry tapeworms involves the gravid segments being passed in the droppings of birds. These segments crawl on the surface of the droppings, releasing eggs that contain a hexacanth embryo. Intermediate hosts ingest the eggs, which develop into cysticercoid larvae. Poultry become infected by ingesting these intermediate hosts. The prepatent period for various poultry tapeworms varies. D. proglottina takes 14 days, R. tetragona 21 days, R. echinobothrida 20 days, R. cestacillus 13 days, C. infundibulum 15 days, C. digonopora 20 days, and Hymenalepis spp. 20 days. Tapeworm infections are more common in free-range birds due to their access to intermediate hosts. Intensive farming can also lead to infections. The most pathogenic tapeworm is D. proglottina, causing necrosis and hemorrhagic enteritis. Chronic infections can lead to weight loss and dull plumage. Diagnosis of tapeworm infections involves examining droppings for gravid segments and postmortem examination of the intestines. Treatment options include various anthelmintics such as niclosamide, praziquantel, and flubendazole. Control measures include maintaining hygiene and using insect growth regulators. Dogs can be infected by several types of tapeworms, including Dipylidium caninum, Taenia hydaceae, Multiceps multiceps, Echinococcus granulosus, and Diphyllobothrium lautum. Dipylidium caninum, also known as the double-pored dog tapeworm, infects dogs and can also occur in humans. It uses dog fleas as intermediate hosts. The rostellum is retractable and armed with rose thorn-shaped hooks. Each segment contains 2 sets of genital organs, and egg packets contain 30 eggs each. The life cycle of Dipylidium caninum involves gravid segments being passed in the feces or spontaneously leaving the host. Eggs are ingested by larval fleas, where they develop into cysticercoid larvae. Dogs and humans become infected by ingesting infected fleas. Pathogenesis of Dipylidium caninum depends on the age of the host. Adult worms are usually not pathogenic, but heavy infections can cause abdominal pain and diarrhea. Gravid segments leaving the intestines can cause irritation around the perianal area. Diagnosis involves examining feces for egg packets, and treatment includes various anthelmintics. Taenia hydaceae infects dogs and wild carnivores, using domestic and wild ruminants as intermediate hosts. The rostellum is armed with 2 rows of hooks, and adults can grow up to 500 centimeters. Each segment contains a single set of genital organs, and eggs contain a hexacanth embryo. The life cycle of Taenia hydaceae involves eggs being ingested by intermediate hosts, where they hatch and release hexacanth embryos. These embryos migrate to the liver and develop into cysticercae. Dogs become infected by ingesting infected meat or offal. The prevalence of Taenia hydaceae infection is high in sheep but low in other hosts. The migration of cysticercae in the liver causes hemorrhagic and fibrotic tracts. Heavy infections in lambs can cause traumatic hepatitis. Diagnosis involves postmortem examination of intermediate hosts. Taenia multiceps is a parasite that primarily affects dogs and wild carnivores, residing in the small intestine. It also infects sheep and goats, causing a disease known as git or staggers in sheep. The metacestode stage, coenurus cerebralis, affects the brain and spinal cord, leading to severe symptoms. The life cycle of Taenia multiceps begins when eggs are ingested by the intermediate host, hatching in the small intestine. The oncosphere penetrates the intestinal wall and reaches the brain and spinal cord. The larval stage matures in about 8 months, and the final host acquires infection by ingesting the infected meat. This diagram illustrates the life cycle of Taenia multiceps, showing the stages from egg ingestion to the development of coenuris cerebralis in the brain and spinal cord of the intermediate host. Pathogenesis of Taenia multiceps varies between hosts. In dogs, adult worms are not pathogenic, but in sheep, the larval stages cause severe symptoms like acute meningoencephalitis. The location of the cyst in the central nervous system determines the specific neurological symptoms. Diagnosis of coenuris cysts in sheep is typically done through postmortem examination and X-rays. However, treatment is considered uneconomical in sheep. Echinococcus granulosus, also known as the dwarf dog tapeworm, affects various mammals, including humans. The larval stage, known as the hydatid cyst, can cause significant health issues. The life cycle of Echinococcus granulosus involves the ingestion of eggs by intermediate hosts, where they hatch and penetrate the intestinal wall, reaching the liver and lungs. The cysts develop slowly, taking several months to mature. Hydatid cysts, which can grow up to 10 cm in diameter, contain multiple protoscolices. These cysts can cause severe health issues if they rupture, leading to anaphylactic shock. In dogs, adult tapeworms are not pathogenic, but in humans and other animals, the larval stages can cause severe health issues. The clinical signs depend on the location of the cysts. Diagnosis of Echinococcus granulosus in dogs involves examining feces for eggs, while in humans, various tests like Cassoni's skin test and ELISA are used. Treatment in dogs includes the use of arecoline hydrobromide to expel adult worms. Treatment and control of tapeworm infections in dogs and humans involve various methods, including surgical removal, medication, and maintaining hygiene. Public education is crucial to prevent infections. Taenia saginata, a tapeworm affecting humans, resides in the small intestine. The larval stage, Cystocircus bovis, develops in cattle, leading to a condition known as measly beef. The life cycle of Taenia saginata involves the release of gravid segments in the feces, which are ingested by cattle. The larvae develop into Cystocircus bovis in the muscles, and humans acquire the infection by consuming undercooked beef. Pathogenesis of Taenia saginata in humans includes abdominal pain and digestive issues. In cattle, the infection is usually asymptomatic but can cause muscle stiffness. Diagnosis involves examining feces for eggs and treating with medications like niclosamide. Taenia solium, another tapeworm affecting humans, also uses pigs as intermediate hosts. The larval stage, Cystocircus cellulosae, causes a condition known as measly pork. The life cycle of Taenia solium involves the ingestion of eggs by pigs, where they hatch and develop into Cystocircus cellulosae in various tissues. Humans acquire the infection by consuming undercooked pork or through autoinfection. Autoinfection with Taenia solium occurs when eggs are released from gravid segments and hatch in the intestine, leading to cysticercosis in various body tissues, including the brain and eyes. Pathogenesis of Taenia solium varies between pigs and humans. In humans, the larval stage can cause severe neurological symptoms, including neurocysticercosis, which leads to seizures and other neurological issues. Diagnosis of Taenia solium in humans involves stool examination and imaging techniques like MRI and CT scans. In pigs, post-mortem examination helps identify the presence of larval tapeworms. Dipylidium lautum, also known as the broadfish tapeworm, affects various fish-eating mammals, including humans. The intermediate host is typically a crustacean, and the larval stage develops in fish. The life cycle of Diphyllobothrium lautum begins when eggs are passed in the feces of the host. These eggs are light brown, unembryonated, and have an operculum. In the environment, the eggs develop into chorocydium, a six-hooked oncosphere with a ciliated embryophore. The chorocydium hatches and swims in the water, where it is ingested by the first intermediate host, typically a cyclops. Inside the cyclops, the chorocydium develops into a procercoid in about 3 weeks. When fish ingest the infected cyclops, the procercoid develops into a plerocercoid within the fish's muscles. Humans or other definitive hosts acquire the infection by eating raw or undercooked fish containing the plerocercoid. The prepatent period is approximately 4 weeks. Pathogenesis of Diphyllobothrium lautum includes mild pathogenicity in dogs and more severe effects in humans, such as tapeworm anemia. This condition can lead to nonspecific abdominal symptoms, glossitis, and macrocytic hypochromic anemia due to vitamin B12 deficiency. Diagnosis is based on clinical signs and fecal examination for eggs. Treatment options include praziquantel, niclosamide, and quinacrine. Preventative measures include avoiding the consumption of raw fish. Cestodes lineatus belongs to the order Cestoideidea and affects various hosts, including dogs, cats, and wild carnivores. The life cycle involves 2 intermediate hosts, the first intermediate host, such as orobotid mites or coprophagus beetles, and the second intermediate host, which can be amphibians, reptiles, birds, or mammals. The metacestode stage includes cysticercoid and tetrathyridium. Morphologically, the adult worm is small to medium-sized with a scolex that has 4 unarmed suckers. The genital pore is located on the mid-ventral line, and the ovary is bilobed. The uterus in the gravid segment is replaced by a par-uterine organ, and there is no separate uterine pore. The life cycle of Cestodes lineatus is not fully understood but is based on experimental infection. It requires 2 intermediate hosts. The first intermediate host, such as orobotid mites or coprophagus beetles, harbors the cysticercoid stage. When these are ingested by the second intermediate host, the tetrathyridium stage develops, mainly in the peritoneal cavity. The tetrathyridium reproduces asexually by longitudinal splitting. The final host acquires the infection by ingesting the second intermediate host. The prepatent period ranges from 16 to 20 days. Pathogenesis of Cestodes lineatus shows that adult worms are generally not pathogenic. However, heavy infections can cause severe diarrhea in humans. If dogs act as the second intermediate host, the tetrathyridium stage can cause peritonitis and ascites. Treatment is similar to that for Dipylidium caninum. Thank you for watching. Please share, like, and subscribe to the GNP Sir Classes official channel for more informative content.